Hi, it's Dr. Phoenix. Today we're going to be looking at lipids as we continue our study of biochemistry. Lipids are our fats and oils and are similar in chemical composition to carbohydrates in that they do contain carbon, hydrogen, and oxygen just as carbohydrates do. However, the ratio of carbon, hydrogen, and oxygen is not in a 1-to-1 ratio as we saw before. Lipids tend to be insoluble in water. They don't mix well with water. And the reason for that is because they are not polar. They don't have relative charges within their molecule. Lipids contain many more carbon-hydrogen bonds than carbs do. And as we mentioned earlier, the energy stored in a molecule is basically stored in the bonds. So it would make sense that if there are more carbon-hydrogen bonds, then lipids would have more energy stored in them than carbohydrates do. And when we get to the actual number of calories that are stored in lipids, you'll see that it is much more. The general example of lipids are fats and oils, as I just mentioned. We can also add waxes to that. Some of the specific examples, as we'll see, are triglycerides, what's known as phospholipids, and steroids. Lipids serve many functions within the body. They are a long-term energy storage molecule. We do use glucose or carbohydrates as our primary energy source, but we store fat for long-term use. Many of our organs are surrounded with fat, and so in that regard, lipids can be used as a cushioning or a protective function. Lipids are also stored directly under the skin, so it's good for insulating us against the cold. Some lipids are used to build hormones, so they have a regulatory function. And there's a type of lipid that we'll look at here in a moment that is the chief component of our cell membranes. And so it serves as a very important structural function. All right, now the monomers of lipids is a little bit in question because depending on what book you read, some will say that lipids do not contain monomers, and technically that's correct. The definition of a polymer would be a large molecule that is built from smaller monomers that are covalently bonded together, and that's where the slight difference is. In lipids, these smaller subunits are not bonded together with covalent bonds. But for our purposes, let's call the monomers of lipids triglycerides. These are simple lipids and the smallest unit of lipids. So triglycerides, we'll go ahead and consider the monomers, although technically they don't build polymers by the true definition of the word. Now, triglycerides are built from two basic components. First, there is what's known as a glycerol backbone. And if you look at a triglyceride as a letter E, it's the side of the letter E. Glycerol has 3 carbons in it. And then there are 3 what's known as fatty acids that come off of the glycerol backbone. All right, so the components of a triglyceride are glycerol and fatty acids. Now, when you look at these fatty acids, let me go back, you can see that they're not identical, and these fatty acids can all be different in some ways. The basic differences, however, are that some are considered what's known as saturated fatty acids, and some are considered unsaturated fatty acids. And I'm sure you've heard those terms before. What makes something saturated or unsaturated? If you look at the carbon backbone of this fatty acid, you'll notice that there are only single bonds between the carbons. As a result of that, since a carbon atom can bond with 4 other atoms, each carbon will have hydrogen on both sides of it as we go down the chain. We would say that this carbon chain is saturated with hydrogens. That's where the word saturated comes from. So there's no double bonds, there are only single bonds here, and as a result, the carbon chain is saturated with hydrogens. These are the kind of fats that or fatty acids that we tend to see in animal sources, animal fats. And you'll notice that this is a very straight chain, and that is exactly the way it is in real life. As a result, that these straight fatty acid chains can stack very, very densely together. And so that's why at room temperature these tend to be solids. The unsaturated fatty acids are called that because if you look at this carbon backbone chain, you'll see that there are places where some of the carbons are bonded together with double bonds. Now when that happens, remember carbons can have a total of 4 bonds around them. If we look at this first carbon that's in green, we'll count the bonds. 1, 2, 3, 4. So there's no place for a hydrogen here, and the same thing with this carbon, there's no place for a hydrogen here. So we would say that this fatty acid chain is unsaturated. It is not full of hydrogens. The reason for that is because of these carbon-carbon double bonds. These types of fatty acids tend to be plant sources, and at room temperature they tend to be oils. Reason for that, these fatty acids are crooked. You can see the bend here. They don't stack as densely together, and so they will therefore be less dense at room temperature, or they'll be a liquid. The second lipid that we're going to look at are known as phospholipids. Phospholipids are the chief component of our cell membranes, and phospholipids have a very different property than triglycerides. Remember we said that triglycerides are basically insoluble in water. They do not mix with water. When you mix oil and water, you know that they completely separate. Phospholipids are quite different than that. If you look at the structure of a phospholipid, you'll see that they are similar to a triglyceride in that they have a glycerol backbone and they have 2 fatty acids rather than 3. In place of that third fatty acid, they have what's known as a phosphate containing group, or a phosphate head. Now what's interesting about that is that that phosphate head is what's known as hydrophilic, or water-loving. And the reason for that is because this portion of the molecule is what's known as polar. It has a charge. You can see a little negative sign there and a little positive sign there. And since water is also polar, this portion of the molecule is attracted to water, so it loves water. However, the fatty acid tails, as we've seen already, are hydrophobic. They hate water, and they do not mix with water. Well, how does this work? By the way, when we look at fatty acids, we tend to draw them like this, like a little lollipop with 2 sticks on it. The circle is the polar head, and these are the 2 fatty acids. So when you take fatty acids and you put them in water, they will arrange themselves in one of 2 ways. They might form a circle like this, which is called a mycel. And notice how all the phosphate heads are facing the water, and the fatty acid tails that don't like the water are pointed to the inside, away from the water. The other way they can arrange themselves is in what's known as a bilayer, a phospholipid bilayer. And you can see that the phosphate heads again are facing the water and facing the water, with the fatty acid tails away from the water. This is how the cell membranes are arranged. Right? There's water inside the cell and there's water outside the cell, and the phospholipid bilayer of the cell membranes forms this type of an arrangement. The third type of lipid we're going to look at is known as a steroid. Steroids, instead of forming these long carbon chains, tend to form 4 rings. These are carbon rings. And these types of molecules tend to be regulatory in function. Cholesterol is an example of a steroid molecule. This is cholesterol. And all of these other examples of steroids, bile salts, vitamin D, our sex hormones, and adrenocortical hormones, are basically constructed from cholesterol as its basic building source. In the lab, we're going to do a test to determine the presence of lipids, and it's a very simple test. It's called the brown paper bag test. When you put a lipid on a brown paper bag and let it soak in, even after it dries out, it's going to leave what's known as a translucent stain. You can hold that paper bag up to the light and sort of see through it. Whereas if you put something like water on it, even if the water soaks in, it will dry and it will leave no stain whatsoever. So you can very quickly tell that there was some sort of a lipid or oil or fat in a substance if it leaves this kind of a stain. Some miscellaneous facts about lipids. We've already mentioned that they're insoluble in water. They don't dissolve or mix with water at all unless it's a phospholipid. And the number of calories in lipids are 9 calories per gram. We saw carbohydrates were 4 calories per gram, so there's roughly double the amount of calories in a lipid as compared to carbohydrates, and that again is because of the increased number of carbon-hydrogen bonds. Okay, I hope that's helpful. Please send me any questions that you need help with over my big campus, and I'll get back to you as soon as I can, or bring your questions to class. Thank you so much. Have a great day.